Again, welcome to SRA Africa webinar. Today, really, we have an outstanding speaker, Dr. Aina Edogun, and let me share with you one slide. Tell us some information about Dr. Aina Edogun. Can you see the slide?
Yes, we can see it. And please, if you're not speaking, can you mute yourself? Thanks.
Thank you. Oh, yeah.
Dr. Aina Edogun, Edogun, PhD, is a professor of molecular and cellular toxicology with special focus on aquatic toxicology at the Department of Zoology, University of Ibadan, Nigeria. Her research addresses issues of impact/risk assessment, the functional and regulatory aspects of differentially expressed genes, proteins and enzymes, and the general stress of contaminants, including endocrine modulators in wildlife species and fish reproductive physiology. Professor Edogun received a BSc in zoology, MSc and PhD in aquatic toxicology from University of Ibadan, Nigeria, and has since then initiated several ongoing active collaborations with universities in Norway, USA, Italy, and South Africa. She has pioneered research into the development and validation of molecular techniques for understanding the molecular and cellular basis of endocrine disruption and obesity in aquatic organisms in Nigeria and other African countries. Her commitment to cutting-edge research and capacity building in molecular and cellular toxicology for sustainable academic and environmental management in Nigeria and other African countries resulted in her receipt of the following international awards and recognition: Global Senior Scholar of the Society of Toxicology, SOT, 2018. Seeding Labs Instrumental Access to Department of Zoology for provision of equipment to universities in developing countries, 2018. African Representative on the Society of Toxicology and Environmental Chemistry, SADC, Global Omics Group, 2018. She was the featured scientist on Thermo Fisher Scientific Apple podcast on Science with August, highlighting the impact of equipment donation on the development of scientific research in Africa. She is passionate about mentoring of the teaming young academic colleagues in Nigeria and has leveraged on her numerous international collaborations to ensure the success of several young academic scientists at the graduate level, especially in training and completion of the molecular aspects of some of her PhD students' research with publications in high-impact journals and postdoctoral positions on completion of their PhD. Professor Edogun is a member of several learned societies and subgroups including Society of Toxicology, SOT, Toxicologists of African Origin, TAO, Women in Toxicology, WID, Reproductive and Developmental Toxicology, RDTSS, Society of Environmental Toxicology and Chemistry, SADC, Pollutant Responses in Marine Organisms, PRIMO, Coordinator, Organization for Women in Science in Developing Countries, OWSD, Ibadan branch, and is a founding member of the Forum of Nigerian Toxicologists, FONTOX. Professor Edogun is the immediate past president of Toxicologists of African Origin and has recently been appointed the West African coordinator of the Society for Risk Assessment, SRA, Africa. She has authored over 80 scientific publications in highly rated peer-reviewed journals and is a reviewer for several journals.
Please join me to welcome Dr. Aina Edogun. Was you able to hear the audio of the slide?
Yes, we were, but there is one participant, yes. Del 2021 needs to mute their phone because it is overriding everybody's.
Sure. Del 2021, please mute yourself. I can mute. Okay. I muted him. Okay, now because we are really we have a lot of guests this afternoon from Egypt, from Nigeria, from Cameroon, from USA, and from South Africa, from many other countries. So this morning we recorded the presentation with Dr. Edogun. So now, and she's here with us. So I will play the recording which it take about 30 minutes. And after that, we will engage in Q&A with Dr. Edogun. So let me share my screen again.
Good day, everyone. I am Aina Edogun of the.
Are you able to see the screen?
No, not yet.
Not yet? Okay. So let me, 1 second here. Okay.
Just open the slide presentation.
Yeah, because it's a recording, Dr. Muhammad.
Oh, okay.
So let me try again. Okay. Can you see it now? Can you see the screen now?
It's
Yeah,
visible.
we can see. Okay.
Good.
All right.
Wonderful. Thank you.
Good day, everyone. I am Aina Edogun of the Department of Zoology, University of Ibadan, Nigeria. I will be speaking on emerging trends in aquatic pollution monitoring and implications for species biodiversity. Ecosystems and human systems are influenced by multiple factors like pollution, as is linked to climate change, and byproducts of resource consumption. Understanding these suite of simultaneously occurring environmental modifications is essential to develop effective response strategies. Multiple environmental stressors interact to produce more than just additive impacts. This implies that stressors, when they interact, may lead to accumulation that increase or become more complex interactions in natural environments. A wide array of chemicals are present in everyday activities like prescription drugs, disinfectants, preservatives, find their way to the aquatic environment and have been associated with various anthropogenic sources like municipal wastewater treatment plants and septic systems. They are collectively categorized as contaminants of emerging concern. They represent a shift in traditional thinking because they are produced by industries and are dispersed to the environment from domestic and agricultural uses. These chemicals influence ecosystems and human systems. The pertinent question is, why evaluate contaminant levels in aquatic environments? Aquatic environments are unique as the ultimate habitat for diverse fish species. They are the ultimate sinks for a complex mixture of largely untreated effluents that contain chemicals from industrial and domestic uses. For Africa, they are natural sources of domestic water supply. As such, when we measure aquatic ecosystem health, it is crucial for measuring or evaluating human health. The multiple sources of pollutants in all ecosystems have one final destination, the aquatic ecosystem. Pesticides, antifouling agents, and sewage, household products containing chemicals for washing and cleaning, like phthalates, phenolic compounds, industrial and pharmaceutical products like non-steroidal anti-inflammatory drugs, anti-malarial and birth control pills, all have negative effects on aquatic life. A wide array of chemicals from industry, agriculture, and domestic sources are classified as endocrine disruptors. These include industrial chemicals like dioxins, plastics and their additives, pharmaceuticals, and metals. They show enhanced resistance towards biodegradation, they are toxic, they exert estrogenic effects, and bioaccumulate in aquatic organisms. A cursory evaluation of pollution trends in the aquatic environment, when viewed within the context of species biodiversity, can be summarized as the burden that an industrialized technological society puts on the natural world. In other words, what we have given to the environment is giving back to us. For Africa and many other developing countries, the application and use of molecular biological responses for monitoring ecosystem health is limited. The huge migration of industries into Africa and Nigeria because of poor environmental protection laws and sparse data on environmental management, makes enforcement of environmental laws difficult. With this pretext in mind, we carried out an overview of highest occurring field-based ecotoxicological studies in Africa, and only 5 countries had published articles, with our research group in Nigeria leading the forefront for providing information on food safety that targets the vulnerable subpopulation, women and children, using molecular tools. A wide array of contaminants are present in the aquatic environment, and they belong to 2 classes, legacy and emerging contaminants. Majority of these contaminants are non-biodegradable or slowly degrading. They are persistent, toxic, and they accumulate in aquatic food chain, which implies a negative effect on ecology and human health. One pollutant that has attracted societal and scientific concerns is tileth esters. PEs are widely used in plastic production in Nigeria, and they have been listed as possible human tumor and cancer promoting agents. We have carried out extensive studies on tileth esters in Nigerian surface waters and reported high tileth ester concentrations in water, sediment, and fish of municipal water supply lakes and lagoons. 4 classes of PEs were detected, and levels were comparable with industrialized countries like Taiwan and China, indicating widespread pollution. PEs have been linked with increased incidence of cancers, metabolic disorders like diabetes. These are some of our published reports on tileth esters in lagoons and lakes in Nigeria. Toxicological responses of organisms to environmental contaminants depends on the characteristics of the compound and on biological factors. We have evaluated mechanisms of contaminant toxicity on measurable toxicological endpoints like steroid hormone precursors, oxidative stress, and phase 1 and 2 biotransformation enzymes. Toxicophysiological responses by biological organisms to contaminants may lead to overt morphological change with clinical manifestation. For all these responses, there's a molecular basis, and molecular responses serve as early warning signals of an impending physiological shift in homeostasis, development, and reproduction of organisms. Ogun River is one of the major Nigerian rivers used for domestic water supply and fisheries. We evaluated gross liver pathology, hepatic transcripts of oxidative stress enzymes in 4 sites along Ogun River that had varying anthropogenic activities. We observed 50% prevalence of hepatic tumors in tilapias from Ogun River in their liver, and this resembled precancerous tumors in the English sole. We reported decreases in steroid hormone precursors, the cholesterol, that paralleled increases in toxicological and oxidative stress responses. Chemicals like PHEs, PCBs were detected in high concentrations in Ogun River, which implied that contaminants may modulate steroidogenesis by targeting steroid hormone precursors. We concluded that fish adaptation may have significant consequences on cellular and biochemical processes that regulate growth, development, and reproduction of fish in Ogun River, and this may have probable human health consequences. Aquatic sediments are a significant source of contaminants affecting endocrine and developmental systems. Contaminants in sediments interact with organisms using various pathways, like diffusion at the water-sediment interface, resuspension, and trophic transfer. We collected sediments from Lagos Lagoon, Nigeria, extracted polar, elutrate, and non-polar fractions, and evaluated toxicity and aryl hydrocarbon-mediated toxicity in mammalian and fish cell lines for 48 hours. All extracts modulated phase 1 biotransformation responses, showing complex chemical mixtures in extracts. The non-polar fraction was most toxic, with increases in CYP1 activities for all extracts and MROD. EROD was, however, inhibited. Our earlier reports showed that Lagos Lagoon sediment contained lindane, 4-isononylphenol, PHEs, and PCBs. We concluded that sediment contained contaminants that induced endocrine disruption and altered energy homeostasis in aquatic life in Lagos Lagoon. Some of our published reports on toxicological responses of organisms to contaminants in different Nigerian environments. Environmental pollution represents an issue of serious scientific and societal concerns in developing countries to ensure sustainable management and protection of aquatic resources. Coastal and inland waters like dams, lakes, rivers, and streams represent one of the most vulnerable aquatic environments due to high contaminant inputs through effluents from extensive urbanization, waste disposal methods, industrial and agricultural activities, and the absence of established monitoring protocols for sustainable environmental management in a developing country like Nigeria. Remote and apparent causes of increases in endocrine-related diseases have been linked to both genetic and environmental factors. While genetic contributions remain relatively unchanged, environmental factors including contaminant inputs is receiving increasing attention in order to understand its extent and role in relation to endocrine-related diseases. In order to address gaps in tropical aquatic organisms monitoring towards environmental protection, we developed and validated field-adaptable molecular biomarker techniques for overt biological effects of contaminants in tropical organisms, proposing that when contaminants with endocrine disrupting properties interact with biological systems, alter gene expression targeting endocrine and neuroendocrine, peroxisome proliferation, biotransformation pathways may occur in aquatic organisms. Endocrine and nervous systems are major communication systems in the body that integrate stimuli and responses to changes in external and internal environment. Both systems are crucial to coordinated functions of highly differentiated cells, tissues, and organs. The endocrine glands synthesize and store hormones and have a sensing and signaling system which regulate duration and magnitude of hormone release through feedback from the target cell. The endocrine system broadcasts hormonal messages to all cells by secreting into the blood and extracellular fluid. Like a radio broadcast, it requires a receiver to get the message. Feedback loops are necessary for regulating the secretion of hormones in the hypothalamic pituitary axis. Investigating how endocrine disrupting chemicals can affect fish gives good insights into effects on human health. We have adopted fish as a relevant model for studying endocrine disruption in vertebrates because the fish endocrine system is similar to those of humans in function, and endocrine disrupting chemicals may produce negative health and reproductive effects using different mechanisms like receptor-mediated pathways and non-receptor pathways that regulate proteins and enzymes. Steroid hormones are derived from cholesterol and they are found in chordates and arthropods. Classical steroid hormones include adrenal glucocorticoids, adrenal mineralocorticoids, ovarian and placental progestogens and estrogens, testicular androgens and neurosteroids. Cytochrome P450 CYP19 is a crucial steroidogenic enzyme that catalyzes the rate-limiting step in the conversion of androgens to estrogens. And steroidogenic enzyme genes are highly conserved in vertebrates and a prime target for EDCs. An important steroidogenic enzyme is the steroided acute regulatory protein STAR, which initiates the process of moving cholesterol from the outer mitochondria to the inner mitochondrial membrane. P450-SEC then converts cholesterol to pregnenolone. These hormone precursors are important rate-limiting step in steroidogenesis and a prime target for EDCs. One can summarize that STAR facilitates cholesterol transfer over troubled water. In order to assess the effects of rubber dam sediment on the steroidogenesis of A295R cells, we extracted contaminants from rubber dam sediment and looked at its effect on steroidogenesis of these cells using 3 extraction methods as described earlier. We assayed for 10 steroidogenic enzyme genes, estradiol and testosterone, and we reported a decrease in STAR, HGMR, and CYP17. The non-polar fraction showed the most severe effects, which indicates ingestion of detritus by fish and may represent possible food web transfer of contaminants. We observed a general biphasic effect of these responses increasing at lower concentrations, and we concluded that detritus may represent possible food web transfer of contaminants to fish with implied human health consequences of biotransformation of and biomagnification of contaminants along the food chain. Some published reports on steroidogenesis in aquatic organisms. Two important biomarkers for detecting fish exposure to estrogenic EDCs are vitellogenin and zonal radiata proteins. Vitellogenin is an important biomarker for fish exposure to EDCs. It is an egg yolk precursor protein that is expressed by adult females of all oviparous species, including fish. And vitellogenesis is an important event controlling oocyte growth through the incorporation of vitellogenin. Egg yolk proteins serve as food reserve for the developing embryo, while zonal radiata, on the other hand, is the envelope surrounding the animal egg. It's a major structural determinant of the eggshell in fish, and ZRP forms the eggshell or envelope that provides physical protection for the growing embryo and allows diffusive exchange of gases. Oba Dam is an enclosed watershed within the University of Ibadan community and receives largely untreated effluents from laboratories, domestic and agricultural sources. We assessed endocrine disruption and intersex in Oba Dam and correlated this with gonadohistopathological changes in tilapias from Oba Dam. We assayed for chemicals which range from mercury to arsenic to organotines, phenolic compounds, PCBs, and we found a high prevalence of intersex in Oba Dam, with male fish consistently expressing higher VTG and ZRP transcripts than females in non-reproductive months. We recorded a striking case of endocrine disruption in a supposedly male tilapia in Oba Dam with 2 pairs of testes and a pair of ovaries. We concluded that the measured contaminants elicited severe endocrine disrupting effects in fish from Oba Dam, Nigeria, giving us like a cause-effect scenario, and this may have severe consequences for growth and reproduction of fish in Oba Dam with implied human health consequences, because Oba Dam is used as a source of domestic water supply for the university community. In another study, we assessed the exposure of breeding crocodiles to endocrine disruptors in their natural environment in Le Croc Farm, which receives sewage treatment plant effluents from the Crocodile River in Limpopo. Now, Le Croc Farm is located in Pretoria in South Africa. We carried out contaminant analysis using water samplers fitted with membrane and sorbent parts. We observed a wide array of contaminants, including anti-epileptic drugs, in water and in a number of endocrine disrupting chemicals that paralleled slightly elevated vitellogenin and zonal radiata transcripts in male crocodiles. And we concluded that contaminants in water caused severe endocrine disruption in captive crocodiles in this farm. 57% of eggs in that season were infertile, showing non-viable eggs. These are some of our published articles in aquatic organisms exposed to endocrine disrupting chemicals in their natural environment in Nigeria and South Africa. Obesogens are natural or synthetic environmental chemicals that can interfere with the nuclear hormone receptors, promoting obesity by increasing the number and size of adipocytes, changing the amount of calories burned at rest, and shifting energy balance to favor calorie storage. We applied the obesogen phenomenon that has been well documented in humans to fish by looking at the peroxisome proliferator activator receptor pathways and its response or modulation by environmental contaminants, especially endocrine disrupting chemicals that are classified as obesogens in fish. In order to understand how fish in contaminated water showed good growth and body condition, we assayed for PPARs, receptors, and biotransformation responses and related this to condition factor in tilapia species from Ogun River, Nigeria. For fisheries biologists, the Fulton's condition index is considered a reliable physiological index of fish growth and health status, such that fish with condition factor greater than 1 is considered to be in good health. For fish that we collected from here, we observed that fish had good condition factor that was greater than 2, and a number of chemicals were assayed from these environments, including PAHs and PCBs. We reported that chemical modulation of P powers and C pyrethroids in fish from polluted sites may be driving the possible obesogenic effect that we observed here. And this indicates a crosstalk between aryl hydrocarbon receptors and pre-power mediated pathways on metabolic and energy processes in fish, especially for wild fish. We concluded that condition factor may not be a reliable index of good growth and health index in fish in contaminated waters, and unraveled the obesogen phenomenon in fish for the first time. Carcinogenic and non-carcinogenic PAHs were detected at sample sites during the study, and this suggests that PAHs may be potential environmental obesogens and may be activating P-power pathways and driving weight gain as observed in fish collected during the study. Some of our published articles on endocrine disrupting chemicals as obesogens in fish and other aquatic resources from Nigerian waters. The ability of organisms to biologically transform pollutants represents significant steps in the metabolism and elimination of chemical agents in living organisms. Biotransformation activities are divided to phase 1 and 2 reactions, and the biotransformation pathways include a battery of genes that are controlled by the aryl hydrocarbon receptor and activated by various endogenous and exogenous compounds. We went further to investigate biotransformation and oxidative stress responses in captive Nile crocodile from Le Croc Farm in Pretoria, based on the reports of endocrine disruption in these captive species. We observed that male crocodiles produced higher biotransformation and oxidative stress responses than females, with higher CYP1, 2, and 3 isoforms, and they had slightly higher levels of antioxidant systems, catalase, GPX, and MDA for males. We were able to correlate these biological variables with contaminant profile, showing a cause and effect relationship in captive crocodiles from these polluted environments. These are some of our published articles on biotransformation responses in aquatic organisms. Plastic pollution is an emerging scientific and societal concern, especially in the 21st century, because of the invention of the single plastic items for major human activities, generating tons of waste from plastic. And there's widespread contamination in the air and water and soil environments because of the contaminants associated with plastics. It has been estimated that by 2050, there will be more plastics in the ocean than fish. This requires concerted advocacy on a global scale to reduce plastic pollution. And this advocacy is gaining momentum in developed countries using the 3R approach: reduce, reuse, and recycle. But such management practices is non-existent in developing countries like Nigeria. Organizations in developing countries are making concerted efforts to ensure the ban on single-use plastic items and towards protection of the aquatic environment. But in Nigeria, such practices are still not developed. We have in Nigeria reported ubiquitous application of PEs as plasticizers, constituting the bulk of plastic production in Nigeria. Phthalate esters that are common in plastics and BPAs have been listed as possible human tumor promoting agents and carcinogens. They act through the aryl hydrocarbon receptor, regulating downstream events that promote tumorigenesis and carcinogenesis. 80% of plastics in the aquatic environment originate from leaching on land. The breakdown process is slow, and there's limited scientific understanding on their ecotoxicological and human health risks, especially for wildlife and humans. Plastics are not readily degradable, and as contaminant vectors, they leach adsorbed organic pollutants into organisms, thereby compounding bioaccumulation and biomagnification along the food chain. One breakdown product of plastics in the aquatic environment is microplastics. Microplastics consist of plastic particles that are less than 5 millimeters in diameter and are constituted a major part of fish food, especially in polluted environments. Based on this premise, we examined fish stomach for the occurrence of microplastics, and we looked at commercial fish species, about 7 of them, from a municipal water supply lake in southwestern Nigeria. We found plastic in all fish stomach with a higher occurrence in the black jaw tilapia growth. The occurrence of microplastics in fish gut is more worrying because plastic-related contaminants have been detected in human urine, blood, and breast milk, and they have been linked with infertility, early onset of puberty, and as their contaminants have been associated too with development of cancers, and they produce endocrine disrupting effects by mimicking endogenous hormones. Our findings on biological interactions of multiple environmental stressors in aquatic organisms show that contaminants exert chemically induced toxicological responses like endocrine disruption and energy homeostasis that are major concerns in modern ecotoxicological research, with a major consequence of loss of species diversity and collapse of fisheries. These have negative impacts on environmental sustainability. Endocrine disruptors target hormone precursors and have negative implications for cellular processes that regulate metabolism, disease, osmotic adaptation, growth and reproduction, and survival of species. The altered expression of VTG and ZRP in aquatic species has negative consequences for reproduction because this will lead to recruitment failure. The ecological consequences of high VTG and ZRP has been associated with kidney failure and increased mortality rates in aquatic organisms, with reduced fish reproductive capacity and low catch per unit effort. This has direct and indirect consequences on human health. The increases in transcriptional levels of PPARs and C-pitoforms show a chemical modulation of metabolic processes. That is, there's a contaminant-induced crosstalk of aryl hydrocarbon receptors and PPAR pathways, which activated obesity development. This may be an adaptive response for obesogenic infects in fish, but this may be at a greater cost to continuous fish survival. Plastics and microplastics, on the other hand, act as contaminant vectors. They have become prominent in the diet of fish, and their higher bioaccumulation and bioavailability of contaminants to biota makes them prime targets for metabolic disorders to fish, reproductive dysfunction, and this is due to widespread human exposure. The occurrence of significantly high concentrations of legacy and emerging environmental contaminants with endocrine disrupting effects in drinking water and aquatic food resources from Nigerian and South African inland rivers have raised significant public health concerns for humans. The endocrine disrupting effects highlighted in our study may explain reported increases in reproductive anomalies observed in aquatic wildlife and reduction in fisheries yield for both freshwater and coastal communities in these environments. This has raised concerns on the role of endocrine toxicity in the low catch per unit effort of landed fisheries, with direct and indirect consequences for humans that feed on fish and use such waters for drinking. We reviewed over 6,000 articles in describing the state of the science on sources, occurrence, and concentration of endocrine disrupting chemicals in drinking water and aquatic food resources from Nigerian inland waters. A total of 11 EDCs were identified from our study and from Nigerian waters and aquatic food resources, with organochlorine pesticides being the most reported in 54 aquatic food resources. Since agriculture contributed significantly to Nigerian GDP in the first quarter of the year, the significant pesticide usage by farmers may explain high organochlorine pesticides occurrence in food resources. Found a high targeted hazard quotient and cancer risk that were higher in children than adults. The decreased aquatic resources yield may be due to endocrine toxicity, immunotoxicity, reproductive toxicity, developmental toxicity, and carcinogenicity. And this calls for urgent, focused, and strategic intervention with policy formulation towards aquatic food safety and public health. Contaminants in rivers and oceans do not distribute according to national borders. So it is possible that the shores of a nation with strict requirements are polluted from a source in a nation not requiring effective wastewater treatment. Furthermore, tsunamis and cyclones originate from African waters that are largely polluted based on the findings of our studies, and they crest in developed countries. This calls for global concerted efforts at protecting aquatic species biodiversity, aquatic species health, and human health. This shows that aquatic pollution is an international question and cannot be resolved within nation states. The Basel Convention reiterated the need for strict compliance to environmental regulation of pollutants on a global scale. Since contaminants in water are not limited by national and international boundaries, it has become important that developing continents like Africa establish a database of ecosystem health for pollutant inputs into African waters towards achieving global SDGs, especially SDG 14, that is concerned with protection of life underwater. This requires comprehensive biomonitoring database for Africa. We need a paradigm shift from convenience products on a global scale to environmentally friendly products to ensure environmental sustainability, with citizen science as the focus, where we focus on plastic reduction of contaminants from plastic, adopting the 3R approach: reduce, reuse, and recycle. Our regulatory agencies in Africa have been encouraged to adopt an African version of REACH, where we register, evaluate, authorize, and restrict chemicals. This will also help in regulating the large upsurge of industries that are being cited in Africa in recent times. We ensure legal protection of our natural habitats by doing this, and ensure environmental and species sustainability. There are a number of indigenous conservation efforts that are peculiar to Africa that we might need to incorporate into our environmental management practices. Science has to do advocacy and policy and monitoring and enforcement of regulations. There is a need to benchmark important scientific outcomes with interventions and policy formulation for wildlife and public health safety. Epidemiological data and cohort data need to be incorporated into the monitoring of endocrine disrupting chemicals to ensure wildlife and public health. I'd like to acknowledge my research team and my collaborators, Professor Augustine Arukwe of the Department of Biology, Norwegian University of Science and Technology in Trondheim, Norway, has been a major collaborator that made most of our inquiries into disruption of the endocrine system at the molecular level in African waters possible. Professor Francesco Regoli of the Polytechnic University of March, Italy, and members of my research team, Drs. Azubike Chikuka in the Nesria, Nigeria, Dr. Richard Ibo, Damilola Omogwemi, Rosalind, Kola, Biola, Damilare Melda, Okpe, and Paul. Our research has been supported by the Norwegian Research Council through Professor Arukwe's grants, the Norwegian University of Science and Technology, the World Academy of Science, and in recent times we got a grant from the Nigerian Tertiary Education Trust Fund to look at microplastics in the Nigerian aquatic environment and its human health implication. I want to thank you all for listening, and it's time for questions and comments and interactions. Thank you, everyone.
Thank you very much, Dr. Edogun, for this outstanding presentation. Now, I would like to invite my colleague, Dr. Bernard Kadugobi, to lead the questions and the answer part of the webinar today. Dr. Bernard?
Yes, Dr. Kadri.
Yes.
I again join you in thanking Dr. Edogun for the wonderful presentation, captured everything that they have done, and we appreciate that. And now it's time for questions. And if you have any questions, please raise your hand and we'll give you the chance to ask your questions. Let me check if there are some in the chat or so. But the floor is open for questions, please.
Please don't feel shy. Just ask the question. So I have one to start. I have one question. Thank you very much, Dr. Edogun, for this outstanding presentation. You really showed a lot of important data. So how many years it took you to produce this amount of great data?
Thank you, Dr. Kadri. It took a lot of time, maybe like 15 years in total to be able to advance beyond looking at the general body physiology to go to molecular studies. Be between like 15 years on the total.
Wow, that's really great. That's really great. Thank you. I think there is, yeah.
There it is from Amy Rosenstein, who is one of the advisors of SRA Africa. She says, and I quote, "I like your idea of incorporating epidemiological data and toxicology data. Any suggestions on how to do this?" Aina?
Yeah, sorry, network is. Thank you, Amy, for that question. The way we're going in science now, we need to bring some other perspectives into our various studies. And this was what informed our incorporating epidemiological data into toxicology data. My initial training was in fisheries biology before I started with toxicology. So we're bringing biological key events in biological development and interpreting and incorporating into toxicology so that we could explain how systems do this. I would probably want to interact more with you to know what you do so that I can give better advice on how you're going to be able to put that perspective together to make it more meaningful and more interesting to your studies. So we could talk about this further. Let me leave my email address so that can help with further interactions on this.
Okay, thank you, Aina. I see Ngozi Ogugwa.
Hello, good evening, Dr. Adegun. Thank you so much for the interesting talk. I'd like to know, what labs did you use? I asked this question because I've had, as an early career researcher, I've had the challenge of assessing labs to conduct some research work. Thank you.
Okay, thank you. Like I mentioned, most of these research inquiries was made possible because of the ability to collaborate with colleagues that are in developed countries. And this is one of, I'm happy I'm going to tell a little story here, because it was difficult to carry out these studies in Nigeria until I went to SOT meeting in 2013, that Society of Toxicology meeting. And this, I'm going to stress the importance of attending international conferences to our colleagues in the continent. Because when I went for that meeting, I met with Dr. Bernard Gadagui, who was the Toxicologist of African origin president at that time. And he was the one who made the link for the professor in Norway. And we were at that time where we were finding it difficult to assay the vitellogenin gene in Nigeria because of limited equipment. But by the time we were able to collaborate with him, bring our ideas to the fore, we assayed in that particular research inquiry 23 genes targeting different pathways of pollution in fish. So attending conferences, joining societies have a lot of benefits that could really make research go from where we are to where we want to be. So it's important that we take up those kind of opportunities to attend conferences, to join societies. Like we have put out proposals for joining Society for Risk Assessment Africa, which is linked to Society for Risk Assessment, the global body. And there are a lot of resources within these associations that can help to make our science better. Because you know it will cost an arm and a leg in the African continent to do this. But when you get collaborators who have some grants that they can buy into your idea, it makes it more interesting to do science. So I will advise that you join this society. There's a call for SRA Africa today. I'm sure if you join SRA, you meet up with collaborators that can assist in helping you achieve this with your lab. Thank you.
Yes, please. I want to ask a question, please. Yeah, my name is Nagwa Arif from Ain Shams University. Thank you so much for this great presentation.
Thank you. Sorry, I couldn't get that. See, this is the problem. I can't hear the question.
Unmute the voice, please.
Okay.
Bernard?
Yes. Can you hear me now?
We hear you very well.
Yes, I can hear you now.
Yeah.
Yeah, please.
Yeah, okay. I'm just asking about the biomarkers or the molecular marker that you used it in the tilapia fish. That could be possible also for the marine fish or the saltwater fish to detect the pollutants or the toxicology on this fish also, or it's only related to the freshwater fish? Thank you.
Okay. Those markers can be used for marine fish and can be used for any other vertebrates as long as they belong to the vertebrate group. Okay. The specific primers to use for that is available and we can find that out. But like I said, that's why we use fish as a template, because when you look at vertebrates from fish to other animals to mammals, it's the same. Especially when you look at the endocrine system, they have similar outcomes. So you could use those, and there are specific primers that work for specific investigations that we want to make. So
okay.
Yes, it is. Yes.
Thank you. Yeah.
Bernard, you are on mute if you're speaking.
Okay, sorry. Dr. Mohammed Sharif.
Yes, I am very happy about the presentation, and it's a wonderful kind of research and a lot of published research papers about the aquatic toxicology, especially for tilapia and for crocodile, for night crocodile. So I just need to mention that I got my PhD from Michigan State University in aquatic toxicology. And my major advisor was Dr. John Giese, if you you know him, is a big professor, a big guy, and right now he moved to Canada, University of Saskatchewan, and my PhD was in effect of one of the organic phosphate pesticides called chlorpyrifos or dorsban on the night tilapia or Ecrimus niloticus. It was, I was dealing with the acute and chronic toxicity effect of this pesticide on night tilapia, including the early life stages, endocrine damage, and histopathology. And it was published in maybe 1990. And when I went back to Egypt, I was involved in 2 big projects supported by World Bank and USAID, and collaborating with University of Michigan, Ann Arbor, and Texas A&M. It was about the wastewater reuse in aquaculture. And we used to raise some fish, especially Ecrimus niloticus and mullets, and treated wastewater. It was in Suez City, by Suez Canal area. And we compared the quality of fish treated in this treated fish pond sewage water with the other fish from fish farms in Lake Manzala using the raw sewage water for raising fish. And we found our fish as better quality than those fish in the market.
It's too long.
And right now we are working with Dr. Abdelrazak on another project, joint project with University of Maryland. With the we have a problem in Egypt with some fish farms. Really, some fish farms using raw agricultural water, wastewater, I mean, and without any treatment. And so a lot of problems with these fish farms over there in some areas in Egypt, especially in the north areas in Kafr al-Sheikh and Dumyat, with River Nile. Yani. So we need to make some kind of treatment for this raw sewage water and raise this fish in clean and treated wastewater, and then reuse this wastewater after that for agriculture. So I think we can make some kind of collaboration with your efforts, Dr. Jan, and we will analyze everything in this area for sediments and water and fish and even the human being over there, because this practice lasted for about decades. So inshallah we'll start this project I have. I hope, I mean, in summer of 23, so 2023, and we can get your help in this project and this your experience. So with the permission from Dr. Abdelrazak, so it will be a big honor and privilege to work with you in this big project, inshallah.
Inshallah.
All right. Thank you very much. Aina, you have a collaborator. I saw Dr. Biti Sokweolu. She dropped off, or she lowered her hand.
Okay.
Oh.
Hi, Bernard.
Hi.
I thought the time is over, so
yeah, we can give you. We have a few. Yeah, we can give a few more minutes. If you can ask your question, that will be fine.
Yeah, thank you very much for organizing this, Bernard, and the rest. Thank you also to Aina for this beautiful presentation. I'm glad I attended. And I kind of overemphasize the issue of collaboration, but what Aina didn't tell us is her consistent, diligent work and patience, even with our collaborators. Commitment to the collaboration in itself, I think it's important. But my question is a short one to Aina. What will you suggest? You've done excellent work in this space in Africa and in Nigeria in particular, and I know pockets of excellence like this across Africa. What will you suggest for Africa in the short term, medium term, and the long term for attaining this sustainable development goal that we're talking about? Thank you.